Thank you. Our next talk is going to be displayed because the speaker was not able to come here. But yeah, I'm introducing the presentation of Dr. Ting Xu. And yeah, she has done some outstanding research on the perspective on slow brain dynamics, on the perspectives of evolution, development, and cognitive or clinical conditions. And she has done impressive work on yeah, ensuring replicability and reproducibility of the findings. So yeah, we are looking forward to the presentation.
Good morning. I'm Ting Xu from Child Mind Institute. In this presentation, I will talk about how functional brain dynamics change across development in both humans and macaques, and how they are altered across different states of consciousness by comparing the awake and anesthetized brain. I have no disclosures. The brain is a dynamic system. It's continuously active, but this activity is not random. There have been a growing literature studying functional dynamics. Our speaker, Xiao Liu, also has a very nice review about an approach called coactivation pattern, CAPS. The coactivation pattern or the synchronized activation from time to time can reflect the brain network configuration. At a certain point, for example, the peak of the global signal, this coactivation pattern also aligns with increased high gamma activity. And if we study the dynamics, not only at every single time moments, but considering the spatial temporal change across the cortex, we can also recognize these dynamics as the propagations. For example, the quasi-periodic pattern, the QPPs from Sheila Kanhor's group, which capture the recurring spatial temporal patterns across large scale networks, typically repeating every 20 seconds. Another study from Xiao Liu's group identified the propagations traveling along the cortical hierarchy in both human and macaque, and it also recurring aligned with the global signal cycle. A recent study using complex PCA methods to extract the recurrent spatial temporal patterns and identify that those propagations are a mixture of standing and traveling waves. The dynamic patterns captured by this method are also representative, showing strong consistency with the result from a variety of other dynamic analysis techniques. So here we apply this complex PCA method to study the developmental change in brain dynamics from childhood to adolescence. As mentioned earlier, the complex PCA can capture the representative propagations. More importantly, it is flexible in setting the recurrence time length because it doesn't really rely on on a fixed time window. Instead, it calculates the recurrence cycle in this phase domain, for example, from 0 to 2 pi and to 2 pi, basically back to 0 like a clock. So this is especially useful since the duration of this cycle may vary with age during the development. So what we show here are the first 3 components captured by complex PCA. The first component is a spatial temporal propagation along the sensory to the association axis. And the second reflecting the dynamics between the task positive networks, like from the parietal attention to the task negative, like default network. And the third component is spanning from the visual regions to the modal and the insular regions. So each of these 3 dynamic patterns explain more than 10% of the variance, and total, they account for over 60% of the total variance. in both adult cohort and the developmental cohort. The pattern showing high similarity between these two cohorts, and importantly, they also exhibit a great test-retest reliability across sessions within individuals for both groups. So this suggests that dynamic patterns captured by complex PCA are reproducible for both across development and at individual level. Now let's take a look of how these dynamic patterns change from childhood to adolescence. We measure the similarity between each individual's dynamic patterns and the adult reference. Starting at as early as age 8, most children already show high similarity to adult pattern with similarity scores over 0.8 across all patterns of those dots. More importantly, the similarity increase with age for all three patterns, suggesting a progressively refinement of the functional dynamics over this period. The brain's spatial temporal pattern becomes more and more like adults, reflecting this ongoing maturation. We also examined how much time each dynamic pattern dominates the moment-to-moment brain activity. We define this as occurrence rates. So this measure can also show a clear age effect. From childhood to adulthood, individuals increasingly spend more time in pattern SA, which is the propagation along the sensory to association hierarchy, and this pattern becomes more dominant as the brain matures. And by early adulthood, individual also begins to spend more time in pattern spanning between the task positive to task negative axis, reflecting more frequent engagement of attention and cognitive control. In contrast, time spent in the visual to motor propagation pattern decrease from adolescent to into the adulthood, suggesting a shift in the brain's dynamic priorities. So if we now focus on the first pattern, which show the strongest developmental effect, and we can examine the occurrence rate at every single phase of this pattern, and we find more interesting results. There's a positive age association with the top-down propagation phase and the negative association with the bottom-up phase. In other words, as we grow older, the brain tends to spend more time in top-down process, moving from association to the sensory regions, and the less time in bottom-up process. More importantly, when we use this occurrence ratio to predict cognitive performance, we find that top-down phase is a better predictor of total cognitive score compared to the bottom-up phase. So this suggests that increasing dominance of top-down dynamics may play a critical role in supporting cognitive development. And we also observe that each of these dynamic patterns are highly consistent with the functional gradients, which represents the microscale organization of brain based on the static functional connectivity. And in the literature, it has been observed that the order of the static functional gradients shifts during the development around age 11. More precisely, it is the explained variance of the hierarchy gradients increase with age, and the modality gradients between the visual and the motor cortex becomes less dominant. And interestingly, this trend, increase and decrease of this hierarchy and the modality gradients, is also consistent with what we find in the dynamic pattern. Increase of the currents for the pattern SA and decrease for the pattern from visual to motor cortex. So to explore this relationship further, we directly compare the occurrence ratio of the dynamic patterns with the explained variance of corresponding static gradients. So here in the top scatter plot, you can clearly see a positive relationship between the current ratio of dynamic patterns in x-axis and the explained variance of the corresponding static gradients in y-axis. And to test this relationship further, we regress out the dynamic patterns and then recalculate the static gradients. As you can see here, the age effect on the static gradients was significantly reduced after we regress out the corresponding dynamic patterns. So this suggests that the amount of time individuals spend in specific dynamic states play a key role in driving the developmental change we observe in the static functional organization from childhood to early adulthood. In addition, such relationship is also observed across species. Here on the x-axis, it's an explained variance of the static function gradient. And on the y-axis, it's a occurrence ratio of the corresponding dynamic pattern, just here calculated using the coagulation pattern instead of complex PCA. But again, we see the positive relationship, suggesting that static gradient is closely linked to how frequently certain dynamic patterns occur. And it is not just the human, but across species in macaques and mouse as well. And across species, we can also see the differences in occurrence ratio are highly meaningful. We can find the matched dynamic patterns in macaque compared to human, but human spend significantly more time in hierarchy dynamic pattern. So if I can put the conclusion more strongly, this means that the time we spend in these hierarchy dynamic patterns may define who we are, human as human. In contrast, we cannot find the matched pattern in mice with this hierarchy dynamics, sort of like missing in rodents. Now let's shift the gears and look at the dynamic patterns across different states of the consciousness, especially comparing awake and anesthetized condition in macaque. Using the coagulation pattern analysis, we identified the common dynamic modes across awake and light anesthesia to deep anesthesia conditions. However, you can see that the strengths of those modes vary significantly with the consciousness level. By mapping these differences between awake and anesthetized condition, we can decode these differences by mapping the differences onto the human space. And all of these differences regions are strongly linked to language, action, emotion, social cognition, and autobiographic memory. So all of these domains closely tied to conscious experience. And the differences between awake and anesthetized state are not only about the strength of the pattern, but also about the occurrence rate of the dynamic modes. In awake state, animals spend more time in this dynamic pattern along the cortical hierarchy. And it is also very similar for what we observed in human. In contrast, on anesthesia, particularly in the deep anesthesia states, the brain is dominant by the global coagulation modes, a more uniform or less differentiated states, and the occurrence ratio for the hierarchy modes is extremely low. And when we use these coagulation modes to predict the functional connectivity using this regression model, we find that the beta weights had a strong positive correlation with the occurrence rate. So in other words, that the occurrence rate of dynamic patterns defines the static functional connectivity across awake and anesthetized states. And it is both the strength and the occurrence rate of the dynamic pattern determine the consciousness states. Finally, I'd like to briefly highlight our recent work, which will be presented at OHBM, on the developmental effect in macaques, focusing on the early childhoods to juvenile periods from 1 to 3 years old in macaque. It's roughly equivalent to human age from 3 to 12 years old. So in this study, we also identified the robust dynamic patterns using complex PCA approach, reviewing early signature of brain dynamics during this critical developmental window. We also explored how neurotransmitter system supports the developmental change in functional dynamics, especially for pattern 1. And in our analysis, we suggest that both the inhibitory and excitatory, basically the GABA and the glutamate receptor system, play key roles as the brain matures. And this system likely help regulates the shifting balance among the networks that underlies the brain developmental refinements of the dynamic brain activity. So our study on developmental of the functional propagation in human is now on bioRxiv, led by Kyung Sum, who will also be presenting the macaque data at OHBM. And Thomas will give a talk and present a poster on how the 3D neurotransmitter maps were reconstructed, which is the map we used in study the macaque dynamics. I'd like to thank all of those co-authors, the Child Mind Institute, and our funding support from NIH. And finally, thank you for your attention.